everyone, and welcome to Research Recap, brought to you by Arbor Assays. In this series, we dive into fascinating studies conducted by scientists around the globe, exploring how they're using Arbor Assays kits to unlock new discoveries. I'm Cassie Shoemaker, Sales and Marketing Manager at Arbor Assays, and I'm excited to highlight the amazing work being done in labs like yours, share insights, and take a closer look at the tools driving innovation in research. So let's get started. Today, I'm joined by Dr. Geeta Gnanadesikan. Geeta is a postdoctoral research fellow at Emory University, where she is studying capuchin monkey social cognition, cooperation, and the hormone oxytocin. She conducted her graduate work at the University of Arizona in the Canine Cognition Center, where she studied the genetic and hormone contributions to individual differences in dog behavior and cognition, as well as the relationships between kids and dogs. Welcome, Geeta. Thank you so much for joining us.
My pleasure.
I can't believe you got to spend your whole graduate work working with kids and dogs. That's so exciting.
Yeah, it was pretty great, especially the days where I could really just play with puppies and call it work.
Yes, yes, you're very, very lucky. Wonderful. Well, can you tell us a bit about the experimental questions you hope to answer with this study?
Yeah, so I'm going to talk today about a study where we were focused on understanding the relationships between kids and dogs and kind of the interactions between them, and also some hormone components, which is the Arbor assays tie-in. And so very big picture, we were interested in, does human animal interaction stimulate the oxytocin system in children? And some work had been done previously in adults, but never in children. And so then also, can oxytocin pathways explain some of the potential benefits to human animal interaction? So some previous work has shown that human animal interaction can have a variety of both psychological and health benefits, but we don't know a lot about the mechanisms underlying that, and oxytocin is a potential mechanism. And then we were interested in that relationship between children and dogs. And so we wanted to know whether the existing relationship between a child and maybe their own dog versus maybe a therapy dog, whether that would influence any of the things we were observing. And then lastly, a lot of work just focuses on the people, and we wanted to know, well, what about the dogs? Are they also benefiting from this interaction? Are they stressed out by this interaction? And so we had this very large study looking at the kids and the dogs and the behavior and the hormones. So this study was actually run as a clinical trial with 3 conditions. We have the unfamiliar dog condition, where the children are interacting with a dog who they've never met before, which is in contrast to the children interacting with their own pet dog. And then we have a non-social control condition, where the children are engaging in solitary play, so puzzles, Legos, etc. And then for each activity, we collect urine and saliva before the condition and then during and after. And we also administer some surveys at the end. And so what this does is allow us to ask questions about the role of human animal interaction generally, but also the specific bond between the child and their dog. These conditions were usually separated by about a week, and at each session, we collected all of these biological samples that we could then analyze for before and after.
Awesome. That sounds like an incredible study and a great thing that you got to work on as you finished up your graduate work. So you talk a lot about oxytocin and how you had a lot of questions around the dogs and the kids and how that was different than some of the work that's been done in the past and what you were hoping to build on. Can you talk to us a little bit more about oxytocin, maybe why you chose to focus on it over any other biomarkers, if there were other markers that you considered, and why you ultimately chose to stick with oxytocin?
Yeah, so oxytocin is known for its roles in maternal infant care and bonding, as well as for pair bonding between mates. But it's thought to mediate other relationships as well, including maybe between friends or in primates between grooming partners. And then also, really interestingly, human animal bonds. And so in dogs, oxytocin has been shown to increase in both the dogs and their owners when they make prolonged eye contact. But again, the effects in children haven't really been looked at before. And oxytocin also has been proposed as a therapeutic treatment for certain conditions, but it's not really clear whether pharmaceutical administrations are as effective as our natural endogenous oxytocin. So one thing we were interested in was whether the interaction could increase oxytocin in a way that might be beneficial, including for children. And then to complement that, actually, we were also measuring cortisol, which is often talked about as a stress hormone, although in many ways it's more of an anti-stress hormone, right? Because it's secreted in response to stressors as part of the body's coping strategy. And one hypothesis regarding the benefits of human-animal interaction is that the positive experience might actually reduce cortisol levels and kind of serve as a stress buffer. And so here we were looking for maybe a decrease in cortisol as a suggestion that the interaction was lowering stress levels, maybe in the children and the dogs was the hope, and not a source of stress for either one.
Yeah.
And then we were really interested in being able to do both, right? Because while oxytocin is often released in response to positive stimuli, it can also be triggered by stress. And so the cortisol really gave us a context in which to better interpret our oxytocin results.
Yeah. Wonderful. I love the idea of getting a pharmaceutical dose of oxytocin just from playing with my dog.
Yeah.
As you kind of talk through those different biomarkers and why you chose them, can you talk to us a little bit? You presented in the paper about your use of our Arbor Assays Oxytocin ELISA kit. Can you talk just briefly about how you kind of came upon this kit? Was it already in your lab? Did you have to go find one? Any other things that kind of brought you to this kit in particular?
Definitely. Yeah, it was one of the kits that my grad school advisor had used previously. And so it was, I think, the kit that he trained me on in terms of learning to do immunoassays. Um, but there ended up being multiple reasons, both scientific and practical, that we wanted to use the Arbor kits. For oxytocin in particular, I did quite a lot of methods work leading up to this study. It was published elsewhere, um, digging into all the details of how to measure oxytocin, which turns out to be incredibly difficult. And I compared multiple of the commercially available immunoassay kits, and pretty consistently we got the best results with Arbor, which was wonderful. And so combined with some involved extraction methods work, we were able to validate multiple methods of measuring oxytocin that we felt more confident in than some of the existing measurements. And I should mention that that is really important. I think sometimes it's tempting to just dive into a study and use the methods that are out there in the literature, but some of those basic validation steps are always important to do yourself in your own laboratory, and especially important when you're using a new species or matrix or any of that. And so some of that, like laying the groundwork of the methods work, really pointed us in the right direction there on assays and extractions.
Yeah.
And then there were also some practical reasons why we really enjoy using the Arbor kits, and that includes they're very easy to use, they have clear instructions, they tend to have like a little bit of extra volume for buffers and standards. So if you make a mistake, you don't have to throw out the entire kit, which just makes a really good user experience. And I'd also love to shout out the Arbor tech support when I have questions about expirations or kit details, or this went wrong, what should we do? Everyone has always been really wonderful about responding with very helpful advice and suggestions.
Awesome. So please tell us what you found. We can't wait to hear about the kids and the dogs.
So I will start by orienting you to the figure. So we've got our three conditions on the y-axis, and we have a standardized change from the pre-session baseline here on the x-axis. And then we've got facets for the child and dog, and for salivary and urinary oxytocin measurements. So there's a lot going on here. But what do we see? First off, you may notice that we don't actually see that much in the urine samples, except for one decrease in the unfamiliar dog. Overall, we didn't find that the urinary measurements were that informative in this study, and we saw more in saliva. Another thing you might notice is that we didn't find much of the way in the way of increases. So we might have expected that oxytocin would increase through this interaction. And actually what we found was that it mostly decreased over time in all of the conditions, but we saw some interesting changes between the conditions. So we can look at that in a slightly different way. So when we compare the dog conditions to the control condition, we see that salivary oxytocin over the course of the whole session was higher in both of the dog conditions than in the control condition. And that effect is a little bit stronger in the pet dog condition. And then on the dog side, we see an increase in salivary oxytocin in the pet dog condition and a decrease for the unfamiliar dog. So what about cortisol? This is really important for adding context to the oxytocin results. Are the dog conditions perhaps just more arousing somehow? But we find that cortisol stays pretty stable in the pet dog condition and actually drops considerably in the unfamiliar dog condition. So this doesn't seem to be too stressful for children, and interacting with an unfamiliar dog may be particularly effective at lowering cortisol. And I'm not showing you this data, but we found that the effect, this lowered cortisol effect, was strongest for the children who had a stronger reported human-animal bond in their survey data, which was really interesting.
Yeah.
And then on the dog side of things, unfortunately, we can't ask dogs if they enjoy these interactions, but one window we have is looking at their cortisol. And so we find that their salivary cortisol actually dropped quite a bit for the unfamiliar dog and even more for pet dogs. And this suggests that they don't find these interactions particularly stressful, which is really encouraging, I think, both for pet dogs in the home and also for the use of therapy dogs with children.
Perfect. Well, that's really exciting. And I think that that has shown a lot of new things that you've uncovered through these studies, through the dogs interacting with children they know, with children that they're supposed to help, and vice versa for children interacting with a new dog, as well as the interacting, of course, with their own dogs. Again, as we talked about at the beginning, for perhaps that dose of oxytocin, and making sure that no one is stressed at least during this interaction. So as you kind of reflect on the work that you have here and that which you presented in the paper, what are three kind of key takeaways you would like anyone watching this to know? Whether it was something that you learned, a technique or an idea that they could take away, or if anyone even wanted to go back to their own lab and do a similar or related study.
I mean, there's like the scientific takeaways, but I think, every project I do, I learn kind of how to do the science better, and like
Yeah,
the lab work especially. And so I think a lot of my takeaways are kind of practical.
great.
So the first one is just to stop and evaluate and maybe even hone the methods first. And so I kind of alluded to this before, but the details of the assay kit, the extractions, the dilutions, the storage, if you take a moment and do some pilot studies, you can actually use that information to do a better study. And sometimes I think it feels like it's slowing you down, but it's often pays off and pays dividends when it comes to
Yes,
the project itself. And so that's one big takeaway. Another one is that I think it's really great when we are able to, and this comes down to funding often, but to measure multiple analytes when possible. It was something we were able to do in this study that was really valuable, and is not done as frequently as I think it should be, because we know that everything in the body is interconnected, and usually we're so focused in on one thing that we kind of miss some of that context. And so I think our oxytocin and cortisol results kind of show how seeing that context can just help you with interpretability, whether it agrees with what you might have interpreted the first time, or whether it adds a new layer and something else to think about. And then the last one is that different biological matrices reflect different things, and they might be better or worse at capturing different effects. And so in this study, we were able to look at both salivary oxytocin, and also the urinary oxytocin, and we didn't find the same things in both. And on the one hand, that can be a little concerning if you're trying to, understand kind of what's happening biologically, and there seems to be this mismatch. But also we know that these are kind of proxy measures for what's going on in the body, and so different methods may be more suited for different kinds of questions. And I think we need a lot more methods work to start teasing apart what is best for what situation. And so I guess the advice there is kind of a combination of being aware that there are those differences, measuring multiple things when you can, and then also building towards understanding what are the limitations of what samples you're working with. And, you know, a lot of my work has been non-invasive or minimally invasive, but when you can get a blood sample, maybe that will help in certain ways that the urinary analysis doesn't always reflect.
Yeah, yeah, absolutely. Those are really wonderful takeaways, and I think important for everyone to consider as we look at, trying not to upset the animal, of course, that's always our priority, but also, collecting a sample that just has minimal utility is not helpful for moving forward either. So thank you so
Yeah.
much for sharing all of that. And for sharing all of your research with us. I think this has been really wonderful, and I know I've really enjoyed learning what you and your colleagues were able to accomplish with the dogs and the children.
My pleasure.
Um, so thanks everyone for tuning in to this episode of Recap. We hope you enjoyed exploring the exciting research that was done by Geeta and her colleagues. If you have any questions or any follow-ups for Geeta, you can feel free to contact her at the information that's shown here on the screen. If you or someone in your lab has published work using our Arbor Assays kits and would like to be featured in a future episode, please be sure to email us at marketing@arborassays.com. Until next time, have a great day, and we'll see you in the next Research Recap.